name is Bisma Ayyaz and I am from Department of Biotechnology. The topic I am going to present to you today is tyrosine, an amino acid. So let's start it. So let's have a look on the contents first. That is introduction, preparation method, which include industrial synthesis and biosynthesis. And then properties, physical and chemical, both, and then application to how and why we use tyrosine, and then we'll conclude it. So let's begin with introduction. So L-tyrosine or tyrosine, symbol TYR or simply Y, also have an IUPAC name which is 4-hydroxyphenylalanine. It's one of the 20 standard non-essential amino acids by the body makes from another amino acid called phenylalanine. Now, what is essential and non-essential amino acids? Essential amino acids are those amino acids which our body cannot produce, and we have to intake those amino acids through food. And the non-essential amino acids are synthesized in our body through biochemical processes. It's an essential component for the production of several important brain chemicals called neurotransmitters, including epinephrine, norepinephrine, and dopamine. Neurotransmitters are basically signaling molecules secreted by a neuron to affect another cell across a synapse. Synapse is a space between cells through which signals is transformed from one cell to another. Epinephrine and norepinephrine are hormones which are gland called adrenal glands produces, and dopamine is a neuromodulatory molecule that plays several important roles in cells. It is an organic molecules, and it controls our emotions like moods and memory and focus on our movements and effects. Skeletal formula of L-tyrosine. As you can see, it has an benzene ring, and it has an hydroxy group attached to its 5 carbon, and then an amino group, and there is another hydroxy group and an oxygen molecule having a double bond. And then the structure of tyrosine at physiological pH, in which the amino group gets hydrogen from the carboxylic group when the bond breaks. Now the preparation method. First we will discuss the industrial synthesis, that is L-tyrosine and its derivatives L-dopa, melanin, and phenylpropanoids and others are used in the pharmaceuticals, that is medicine, drugs, and dietary supplements and food additives. Food additives which are flavors and several preservatives. Two methods were formerly used to manufacture L-tyrosine. The first involves the extraction of the desired amino acid, which is the tyrosine, from protein hydrolysates. Protein hydrolysates means the breaking of protein using a chemical approach. The second utilizes enzymatic synthesis from phenolics. Phenolics are basically a benzene ring with a hydroxyl group attached. Pyruvate is a molecule which we get through glycolysis process, in which the glucose is broken down into 2 pyruvates molecules and ammonia through the use of tyrosine phenylyase. Advances in genetic engineering and the advent of the industrial fermentation have shifted the synthesis of L-tyrosine to the use of engineered strains of E. coli, that is a bacterium called Escherichia coli. Now let's focus on biosynthesis. In plants and most microorganisms, tyrosine is produced via propionate, an intermediate on the shikimate pathway. Propionate is oxidatively decarboxylated with retention of the hydroxyl group to give p-hydroxyphenylpyruvate, which is transminated using glutamate as the nitrogen source to give tyrosine and a ketoglutarate. As you can see in the diagram, how the bonds are breaking and molecules are combining to form the molecule tyrosine. Mammals synthesize tyrosine from the essential amino acid phenylalanine, PHE, which is derived from food because it is an essential amino acid. The conversion of PHE to TYR is catalyzed by the enzyme phenylalanine hydroxylase, a monooxygenase. This enzyme catalyzes the reaction, causing the addition of a hydroxyl group to the end of the 6 carbon aromatic ring of the phenylalanine, such that it becomes tyrosine. And that's how our body generates or synthesizes tyrosine from an essential amino acid, because tyrosine is a non-essential amino acid. Now let's have a look on properties. Physical properties. Taste. It has when it's in a moderate state, tyrosine results in a light bitter state. Its color is when it is irritating is yellow. It is a non-essential amino acid and it has a polar side group. pH. The pKa, the strength of acid of tyrosine in the excited state is between 4 and 5, as the negative value reflects the formation of tyrosine weak base. Hydrogen bond complex or tyrosinate ion in the excited state at pH 4.63 and 5.51. Solubility. The solubility of tyrosine is largest in DMSO, dimethyl sulfonide oxidase, and lowest in N-propanol. At a given temperature, the solubility of L-tyrosine is greater in DMSO and water than in the other three solvents. Chemical properties. Melting point is greater than 300 degrees Celsius. Boiling point is 314.29 degrees Celsius. It's a rough estimate, not an accurate one. Density is 1.34 kilogram per cubic meter. Refractive index is minus 12 degree, 1 mole per liter, HCl. Flash point is 176 degrees Celsius. Storage temperature, store below positive 30 degrees Celsius. It should be less than 30. And then solubility, a 1 molar HCl, 25 milligram per milliliter. Form is in powder form. Color is white to pale brown, and like I tell you in the physical properties, it is yellow when it is irritated. Water solubility is 0.45 gram per liter at a 25 degrees Celsius, which is a room temperature. Stability, it is stable, but it is incompatible with strong oxidizing and reducing agents. Applications. Phenol functionality. Aside from being a protogenic amino acid, tyrosine has a specific role by virtue of the phenol functionality. It occurs in proteins that are part of signal transduction processes and functions as a receiver of phosphate groups that are transferred by protein kinases. Like protein kinases means the enzymatic processes. Phosphorylation of the hydroxyl group can change the activity of the target protein or may form part of the signaling cascade via SH2 domain binding. A tyrosine residue also plays an important role in photosynthesis. In chloroplast photosystem 2, it acts as an electron donor in the reduction of oxidized chlorophyll. In this process, it loses the hydrogen atom of its phenolic OH group. The radical is subsequently reduced in the photosystem 2 by the 4 core manganese clusters. Photosystem 2 it is included in the light reactions and dark reactions occurs in plants. In light reactions, when there is phosphorylation, which is basically the chemical addition of a phosphoryl group PO3 negative to an organic molecule, and photosystem 1 and photosystem 2 is basically the same thing as in light reactions. It is a diagram of the metabolism, how it is metabolized and converted into different molecules like dopamine, noradrenaline, and adrenaline. Phosphorylation and sulfation. Some of the tyrosine residues can be tagged at the hydroxyl group with the phosphate group phosphorylated by protein kinases. In its phosphorylated form, tyrosine is called phosphotyrosine. Tyrosine phosphorylation is considered to be one of the key steps in signal transduction. Signal transduction means signal transfer and regulation of enzymatic activity. Phosphotyrosine can be detected through specific antibodies. Tyrosine residues may also be modified by the addition of a sulfate group, a process known as tyrosine sulfation. Tyrosine sulfation is catalyzed by tyrosyl protein sulfotransferase TPST. Like the phosphotyrosine antibodies mentioned above, antibodies have recently been described that specifically detect sulfotyrosine. Precursors to neurotransmitters and hormones. In dopaminergic cells in the brain, tyrosine is converted to L-DOPA by the enzyme tyrosine hydroxylase. Tyrosine hydroxylase is the rate-limiting enzyme involved in the synthesis of the neurotransmitter dopamine. Dopamine can then be converted into other catecholamines such as norepinephrine, noradrenaline, and epinephrine, adrenaline, which both hormones are released by the adrenal glands. The thyroid hormones triiothyronine and thyroxine T3 and T4 in the colloid of the thyroid are also derived from tyrosine. These both hormones are produced by our thyroid glands in our body. Precursors to alkaloids. The latex of Papaver somniferum, the opium poppy, has been shown to convert tyrosine into the alkaloid morphine, and the biosynthetic pathway has been established from tyrosine to morphine by using carbon-14 radio-labeled tyrosine to trace the in vivo synthetic route. It is also which means it is also used in the radioactive process in the in vivo synthetic route. In vivo means it is tested inside the human organisms on basically on the whole human or any living being. Role in coenzyme Q10 synthesis. Tyrosine or its precursor phenylalanine is needed to synthesize the benzoquinone structure, which forms part of coenzyme Q10. Precursors to natural phenols. Tyrosine ammonia lyase (TAL) is an enzyme in the natural phenols. As I told you before, phenols are the rings which have an hydroxyl group attached to them. Biosynthesis pathway. It transforms L-tyrosine into p-fumaric acid, precursor to pigments. Tyrosine is also the precursor to pigment melanin. Melanin is a hormone released in our body by pituitary glands, which controls our skin color. Degradation. The decomposition of L-tyrosine synchronized also as para-hydroxyphenylalanine begins with an alpha ketoglutarate dependent transmission through the tyrosine transminase to para-hydroxyphenylpyruvate. The positional description para abbreviated P means that the hydroxyl group and the side chains on the phenyl ring are across from each other. You will see it illustrated below in the diagram in the next slides. The next oxidation step catalyzes by p-hydroxyphenylpyruvate deoxygenase and splitting of CO2 homogentisate 2,5-dihydroxyphenyl-1 acetate. In order to split the aromatic ring of homogentisate, a further deoxygenase, homogentisate 1,2-deoxygenase is required. Thereby, through the incorporation of a further auto molecule, melyl acetoacetate is created. Furthermore, for fumaryl acetoacetate is created by melyl acetoacetate cis-trans isomerase, in which the functional groups are changed in cis-trans manner through rotation of the carboxyl group created from the hydroxyl group via oxidation. This cis-trans isomerase contains glutathione as a coenzyme. Fumaryl acetoacetate is finally split by the enzyme fumaryl acetoacetate hydrolase through the addition of a water molecule. Therefore, fumarate, also a metabolite of the citric acid cycle, and acetoacetate 3-ketobutyrate are liberated. Acetoacetate is a ketone body which is activated with succinyl coenzyme A, and thereafter it can be converted into acetyl coenzyme A, which in turn can be oxidized by the citric acid cycle or be used for fatty acid synthesis. Folaratic acid is also a urinary metabolite of tyrosine in rats. As you can see in the whole process I described to you below in this diagram, in which the tyrosine is changing into different molecules by breaking and combining of bonds. It also has a medical use. Tyrosine is a precursor to neurotransmitters and increases plasma neurotransmitter levels, particularly dopamine and norepinephrine, non-epinephrine, but has little if any effect on mood in normal subjects. However, a number of studies have found tyrosine to be useful during conditions of stress, cold, fatigue, in mice, prolonged work and sleep deprivation with reductions in stress hormone levels, reduction in stress-induced weight loss seen in animal trials, and improvements in cognitive and physical performance seen in human trials. Cognitive means thoughts. Tyrosine does not seem to have any significant effect on cognitive or physical performance in normal circumstances, but does help sustain working memory better after multitasking. Conclusion. Tyrosine is basically an amino acid, a type of amino acid which are the building blocks of proteins. The body makes tyrosine from another amino acid called phenylalanine that is already described above. In the diet, tyrosine can be eaten in dairy products, meat, fish, eggs, nuts, beans, oats, and wheat. The body uses tyrosine to make chemical messengers that are involved in conditions affecting the brain, such as mental alertness. People most commonly use tyrosine in protein supplements for an inherited disorder called phenylketonuria. It is also used for alcohol use disorder, cocaine dependence, and memory and thinking skills, but there is no good scientific evidence to support most of these uses. Thank you so much for your time.